Hi everyone and welcome back to Neuropsychology. So in this chapter we are going to start talking about drugs and hormones, and we will go over how drugs affect our nervous system and our behavior. So we will begin by looking at ways how the drugs are eliminated from the body and how they act at our synapses. In a later video, we will talk about psychoactive drugs, and then we'll group them in 7 groups based on their major behavioral effects, and we will examine their individual responses to addiction. Many drugs are also related to hormones, and many drug-related principles apply to the action of hormones. So that's why in the last topic, we will talk about hormones. Okay, so after a drug has been administered, the body will soon begin to catabolize and try to remove the drug from our body as soon as it can. Diluted drugs are sequestered in many regions throughout the body, including our fat cells. So they are catabolized throughout the body and then broken down in the kidneys, in the liver, and also in our intestines by bile. Then the drugs will eventually be excreted in urine, feces, sweat, breast milk, and exhaled hair. Drugs that are developed for therapeutic reasons or purposes are usually designed to increase chances of reaching its target organ. So for example, if it's a heart drug, it has to reach the heart, and it works to enhance the survival time in the body. So the longer it's active in the body, the longer it will do its job. However, drugs that are eliminated from the body can still remain a problem. They may be re-ingested through food or water by many animal species, including other humans. And some may have effects to fertility or embryonic development and even our physiology. So this problem can be limited by redesigning waste management systems and to remove drug byproducts and how to eliminate these in humans and also in other animals. Okay, so we briefly talked about the blood-brain barrier in one of the first couple of videos. So remember that the blood-brain barrier prevents many substances from entering the brain's capillary network. So in the picture below, where's my mouse? So there. In the picture below, right here, you can see a single really thin layer of flat endothelial cells that make up our brain capillaries. So right here. So the little end feet of these astrocyte cells surround about 80% of the outside surface of these brain capillaries. And astrocytes will help maintain the tight junctions between the fused membranes of the endothelial cells. This will allow healthy substances to pass through these cells in one of each ways. Okay, so let's move on here. So molecules like glucose, amino acids, and other nutrients that are good for us can be carried across the capillary by active transport systems. So for example, specialized protein pumps, such as the sodium-potassium pump, can pump these substances through. And small molecules such as oxygen and carbon dioxide, which are not ionized and they are fat soluble, they can pass through the capillary wall with no problem. So the capillary cell wall in only 3 brain regions do not have a blood-brain barrier. So the pituitary gland of the hypothalamus secretes many hormones into the blood, and other hormones carried to the pituitary gland in the blood triggered by their release. So the pituitary gland is one of them. The absence of the blood-brain barrier at the area postrema of the lower brainstem allows toxic substances in the blood to trigger a vomiting response. And lastly, we have the pineal gland, The pineal gland also lacks a blood-brain barrier and is therefore open to the hormones that modulate day and night cycles this gland controls. So remember that these three areas in the brain do not have a blood-brain barrier. Okay, so let's talk about drug actions in the synapse. So almost all psychoactive drugs have been discovered accidentally. Most psychoactive drugs work by influencing the chemical reaction at the synapse. So understanding this synaptic activity in the brain might help explain both the psychoaffective effect of the drugs and their potential harm to humans. So here in the image, you can see several major events that contribute to synaptic neurotransmission. So each of these sites, a drug can act upon. So for example, neurotransmitter synthesis can take place in the cell body axon and terminal. And this can also affect how the drug works. And then the neurotransmitters, where's my mouse? And then the neurotransmitters may then be stored in storage granules or vesicles until it is released from the terminal's presynaptic membrane. And then when it's released, it can act on a receptor which is embedded in the postsynaptic membrane. And then neurotransmitters are then either deactivated or they're taken back into the presynaptic cell. And the cell has also its ways of degrading a neurotransmitter and removing unneeded byproducts. So, for example, SSRIs, or technically serotonin reuptake inhibitors, they block number 6. So as the word says, serotonin reuptake inhibitors, they inhibit the reuptake of these SSRIs, so they stay in the cleft longer, which means they will be active for longer. Okay, so each component of neurotransmission entails one or more chemical reactions that drugs can potentially influence. So we have two types of drugs. We have agonists and we have antagonists. Agonists are drugs that increase the effectiveness of neurotransmission. An example of this is the SSRI we talked about earlier. Antagonists are drugs that decrease the effectiveness of neurotransmission. So agonists and antagonists can only work in a variety of ways. However, their results are always the same. So let's go over some examples. Let's focus on one neurotransmitter, or acetylcholine. And then we're going to talk about some agonists and antagonists for this neurotransmitter. So the following are agonists and increased effectiveness of acetylcholine transmission. So we have black widow spider venom, nicotine, physostigmine, and organophosphates. So black widow spider venom promotes acetylcholine release to axis. So spiders use this on their prey as it will cause paralysis or death. A spider bite does not contain enough toxicity to paralyze a person, but if you ever get bit by the spider, you can feel muscle weaknesses. Next up is nicotine. Nicotine in cigarettes stimulates cholinergic receptors called nicotinic receptors. Nicotine structure is similar to acetylcholine and fits in the acetylcholine receptors. Then physostigmine, it inhibits the breakdown of acetylcholine. So it is obtained from the African black bean, as you can see in this picture right here, and it was used to poison other tribes. Large doses can be toxic and have similar functions as spider venom. And lastly, organophosphates are found in insecticides. They are very toxic, and it binds to the enzyme that breaks down acetylcholine, and it binds irreversibly. So whenever it binds, it doesn't release. So it will inhibit the breakdown of acetylcholine after it has done its job. So the following two are antagonists. So botulinum toxin and curare. So curare is a poison extracted from a South American plant, and it blocks cholinergic receptors and prevents acetylcholine from having an effect. Large doses can stop movements and breathing. And then botulinum toxin is an acetylcholine antagonist because it blocks the release of acetylcholine. It is used in medicine because it paralyzes the muscle, and therefore it can block twitches. It's also used in cosmetic surgery, and it's used to reduce wrinkles by relaxing the muscles and sometimes reducing pain. Okay, so here we have them again. So black widow spider venom, it promotes the release of acetylcholine, so it's an agonist. And then botulinum toxin blocks the release, so it's an antagonist. Nicotine stimulates the receptors, so it's an agonist. Curare blocks the receptors, so it's an antagonist. And physostigmine and organophosphates, they block inactivation, so they're agonists because they keep the neurotransmitter in the cleft. Okay, so before we start talking about the groups, I wanted to talk about some terms. So one of them is tolerance. And tolerance is a decline in response to the repeated administration of a drug. So here you see it's a really old study that has been done in prison volunteers where they studied tolerance. So this study will never pass regulation anymore, but it has been done in the way past. And we did learn a lot from it. So what the researchers did is they gave the participants enough alcohol every day for 13 weeks to keep them in constant states of intoxication. It's crazy, huh? So yet they found that the participants did not stay drunk for the entire 13 weeks. So in the first days of the experiment, participants showed rapidly rising levels in blood alcohol levels and in behavioral signs after consuming alcohol. After about 15 days, the BAC, or the blood alcohol concentration, and the behavioral signs fell, as you can see in the bottom two graphs, around like 15 days all the way down. So here you see the average blood alcohol level, and here you can see it goes up, 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 the more alcohol they drank every day, and then after 15 days, it will drop. So there are different kinds of tolerance and different things that work on tolerance. So we have metabolic tolerance, we have cellular tolerance, and we have learned tolerance. So let's go over these. So metabolic tolerance results from an increase in enzymes that is needed to break down alcohol in the liver, blood, and in the brain. So a person metabolizes alcohol quickly, so blood alcohol levels are reduced. Cellular tolerance results from adjustments to the activities of the neurons to minimize the effects of alcohol. Behavioral signs of intoxications are low despite high blood alcohol levels. Learned tolerance results from learning to cope with daily demands under the influence. Thus, a reduction of outward signs or behavioral symptoms is seen. Whereas tolerance generally develops with constantly repeated use of a given drug, sensitization increases responsiveness to equal doses of a drug and is more likely to develop with occasional use. So it's kind of the opposite of tolerance. So it produces structural changes in the brain that make the brain more sensitive to a drug. So scientists studied this using rats, and they gave them amphetamine and recorded their reactions. So amphetamine stimulates your dopamine receptors. And every 3 days, they injected these rats and then observed their motor activity. So they observed when they were sniffing, when they were rearing or walking. And it was more vigorous after each administration of the same dose of the drug. So the increased response was not due to them getting comfortable in their test situation. Control rats had received no drug and did not display this behavior. So every time they got the drug, they increased their motor activity. Even when 2 injections were separated by months, the animals still showed increased responses to the drug. Sensitization is a long-lasting thing because it is accompanied by structural changes in the brain that are very long-lasting. So rats that show sensitization to amphetamine, or actually also cocaine, undergo increased dendritic growth and also increased spine density compared to control rats. So in this study, the effects were found in the nucleus accumbens and in parts of the basal ganglia. So you can see that there is an increase in dendritic growth and also in spine density. So the spine density here, you can see there's more little spines on these dendrites compared to rats that were not given the drug. There's less dendritic growth and there's less spines. So in conclusion, sensitization induced by repeated exposure to amphetamine changes the structure of our neurons. Okay, can drugs cause brain damage? So the simple answer is yes. But determining which drugs do so is very difficult. So many natural substances can also act as neurotoxins. For example, caffeine or tetrodotoxin that we talked about earlier that is found in pufferfish. The strongest evidence that recreational drug can use brain damage and cognitive impairment is from studies from the synthetic amphetamine-like drug MDMA. Findings from animal studies show that doses of MDMA approximating those taken by people that take them at festivals and parties resulted in degeneration of very fine serotonergic nerve terminals. In monkeys, they show that this terminal loss may be permanent. So in the left image here, this is a monkey brain. In the left image here, you see the density of serotonin axons in the neocortex of a squirrel monkey. And on the right, you see the squirrel monkey after 18 months of MDMA treatment. And you can see that a lot of these axons are gone. On top of that, MDMA also impairs your memory. Psychoactive properties of cocaine are actually pretty similar, and they also have really bad side effects. Cocaine use is relative to blockage of cerebral blood flow and other changes in blood circulation. So brain imaging studies suggest that cocaine use can be toxic to neurons because a number of brain regions are reduced in size in cocaine users. Okay, this was the end of the video, and I will see you guys back soon where we are going to start talking about the 7 drug types.